I'm going to talk about the role that genetics may play in the future in therapy, but also, most importantly, in our knowledge of mental illness. So, psychiatric genetics has played for many years, actually for a century, an important role in mental health or in psychiatry. It started very early. It started more than 100 years ago, or 100 years ago, in the early 20th century, with family studies that led to twin studies and then adoption studies. They have given us the firm knowledge that there is a genetic basis. Based on that, people then started applying methods that were designed to find genes, like genetic linkage studies in large pedigrees and candidate gene studies in samples of cases and controls. Then past the year 2000, when the Human Genome Project was finished or came to a first stop, people started doing genome-wide association studies, studying millions of markers. And now we are in the era of sequencing. That means trying to find new variants that might play a role. So while in the 20th century, we studied genetics, one polymorphism, one variant at a time, we are now studying genomics. That means the whole genetic variation that is there in the genome. And we have big data, we have large consortia that we can work with. We have powerful molecular tools. The technology has advanced. The statistics have advanced. We now can perform these studies like genome-wide association studies. However, when you do that, when you study so many variants, you have to be very careful about claiming significance. So you have to apply a very stringent threshold in terms of significance. So a finding is only significant when it reaches that small level of that small p-value. But we have now consortia, big consortia like the Psychiatric Genomics Consortium, that help us achieve those large samples that allow us then in turn to produce results at this level of genome-wide significance. And this is just to give you a brief overview of where we stand. Many traits in medicine, like almost everything, has been studied by genome-wide association studies. In psychiatry, most importantly, schizophrenia, bipolar disorder, major depression. And what these plots show, these are called Manhattan plots because they resemble the skyline of Manhattan. That's the idea behind it. So here you see the chromosomes, here you see the p-value, the -10 log, and this is the level of genome-wide significance, this threshold. So you have to cross this threshold so that you can claim significance. And all these dots here, these are markers. They are above that line. So we have several hundred thousand cases of controls. We have more than 400 loci combined findings. They confirm previous findings and also show us novel leads. And we also perform cross-disorder approaches so as to find genetic variation that is responsible for maybe common aspects or for aspects that these disorders have in common. Again, these things have been done now for many phenotypes, many disorders, and it's going on. Actually, now the latest results, you will have many more findings because they put together the largest samples that are available in the world today. But just to give you an idea of what it looks like. Now, you can use these genome-wide association studies and look at each an individual marker at a time. But an individual marker only gives you so much information, hardly any information, because the risk associated with a marker is very small. But you can combine all these markers together and build one score. It's the polygenic profile score. So you can run an analysis in one sample and then in a large sample, and then you can build a score. And then in another sample, maybe you have a sample of people that you test for therapy response, and you want to see, does someone with a high score for schizophrenia, high schizophrenia, polygenic score for schizophrenia, does this person, maybe that's a person with bipolar disorder, does that person have a better outcome in a certain treatment than someone with a higher score? Do they have a worse outcome than someone with a lower score? So you can play then, and you can answer many questions. So this is actually a benefit of having this genome-wide association background. How useful it is in the clinical field, we will see. But I'm personally convinced that in the future, all of us will use these scores like we use liver enzymes or whatever markers. And it will be one aspect of our overall diagnostic procedures. So I would say, while we did not get what we hoped for 3 decades ago, when I started, the idea was that we have 20, 30 genes, and that's it. And they would explain everything, but that's not the case. Well, that was the idea of an oligogenic model. We now clearly know it's polygenic. But I would still say that we have made tremendous progress over the last decade, because we have identified many genes. We have evidence now of polygenicity and can use this, as I said, for polygenic profile scoring. And we also have, which I'm not showing now, evidence of an involvement of rare genetic variation, such as copy number variants, or CNVs, in intellectual disability, autism, and schizophrenia. And this actually can be used in diagnostic workup, in genetic testing, especially in child and adolescent psychiatry. And again, there are many novel leads that warrant downstream analyses. But I would like to go beyond the mere genomics aspect. I think we should explore the use of polygenic scoring to understand etiology, comorbidities, diagnostic overlaps. We should go deeper and more longitudinal in our phenotypes. We should focus on treatment and outcome. We should go beyond just mere genomics, and we should include multi-omics. What do I mean by that? So just the first example of our group together with the group in Switzerland. So here we looked at the impact of polygenic risk scores across the extended psychosis spectrum. So we take polygenic scores derived from schizophrenia samples, and we can show that it also, these scores, or this score predicts schizophrenia in other samples, but also to a certain point predicts risk for bipolar disorder in other samples. The other way around, bipolar disorder predicts bipolar disorder and even schizophrenia in other samples. So you see the connection, you see the joint kind of the overlap. But what we also looked at was whether either score, whether any of these scores would predict also schizotypy. So because people think or describe schizotypy as a personality trait as something that is maybe part of the psychosis spectrum, part of or like a precursor. Well, we could not find any effect of polygenic profile scores on either schizophrenia or bipolar disorder on the phenotype of schizotypy. So maybe, after all, schizotypy may be something totally different and not part of this spectrum. This is just our study. Other studies would have to prove that or disprove that. But I think it gives you an idea of what you can do with polygenic profile scores. Other thing, looking at more clinically relevant phenotypes, like genome-wide, we did a genome-wide association study of age at onset and polarity of onset in bipolar disorder. We, you see, it's many authors. That one of my doctoral students led this initiative, and we wanted to get the data from data on age at onset and polarity of onset, whether you start with mania or depression. We got the data. We hope to get data from all the samples that are in the Psychiatric Genomics Consortium on bipolar disorder. However, it was not as easy as we thought. It took us 5 years to reach out to all these people. And there are like 60,000 samples, but we only got like 15,000 samples with this information for age at onset, and only roughly 7,000 for polarity of onset. Now, I think it's just a shame, because I think we as treating psychiatrists, we know when the depression kicked in or when the mania started, but the information doesn't make it into these databases. So that's a big problem. Anyway, we were able to pull these data together. We performed a study, and we got a genome-wide significant finding in a gene that has been found associated also with metabolic disorders. We didn't find anything for polarity of onset. The samples are just too small. So it gives you an idea that you can actually pull out some information, even with a smaller sample, if it's maybe more homogeneous, the phenotype, age at onset, maybe more precise, or maybe more, it's not just bipolar disorder, it's not just schizophrenia, it's a very refined phenotype. So this gives me hope that if we, in the future, are able to pull together more phenotypes in these samples, we may be able to find more genetic variants. But again, we need more samples. So we couldn't replicate that in the smaller replication sample, but we can demonstrate using the profile scores that even having liability for autism, schizophrenia, depression is also associated with an early age at onset in bipolar disorder, but we need larger and better characterized samples. And the clinical input is very important. So another aspect, longitudinal psychiatric research. Most of the research that we read and talk about in psychiatric genetics is not longitudinal in nature. It is based on samples that were established at one point, cross-sectional, and that's it. One assessment at one time point. But we all know, as psychiatrists, we do not treat just the diagnosis. We treat a course and an outcome. We want to see that a patient doesn't come back. We would like to see a patient succeed in life. We want to see that a patient goes back to work, goes back to school, and so forth. That's about outcome, right? So, and this we can only study if we follow patients longitudinally. So we started a while back in Germany, and with a site in Austria, a study where we had a longitudinal cohort project on schizophrenia, bipolar, major depression. We currently have 1,700 people. And well, it's actually finished, the collection. We had 4 time points. We biobanked them, DNA, RNA, serum, plasma at each visit. And this has been described in this paper. It's the PsyCourse cohort, and you can look that up. There's plenty of opportunity for collaboration. The sample is available for collaborative projects. The sample is described online in detail. All the variables, all the statistics can be looked at. So if you have a question, or if you want to collaborate, if you have an idea of what you want to do, maybe in that field, please feel free to reach out to us. What we have been doing, we have been exploring novel clusters. So instead of saying we just analyze schizophrenia, depression, or bipolar disorder, what we do here is we have clusters, longitudinal clusters, that in the future may be used in other analyses. So instead of talking about, do schizophrenia patients with this medication do better than schizophrenia patients on this medication, we can say, does a cluster A, which includes maybe 70% schizophrenia patients, but also 20% bipolar and 10% depressed patients, does this cluster improve better, and so forth. So it's a more maybe creative way, innovative way of thinking. While we enter the classic diagnostic criteria, we can play. We can change the settings in this longitudinal clustering approach. So that's the power of longitudinal approaches. Another important area for me is pharmacogenetics. In 2008, I started this consortium together with my dear colleague Francis McMahon at the NIH on lithium genetics, because we know that lithium is a great drug. It works and has been working for almost 80 years now, but we don't really know why. And so we wanted to bring together researchers that are interested in that topic and founded the Consortium on Lithium Genetics. It was small in the beginning, but it has grown. A dear colleague of mine from Cagliari, Mirko Manchia, was here instrumental in putting together that paper where we described the phenotype that we use to assess lithium response retrospectively, which is a scale developed by Martin Alda and Paul Groff from Canada. And so, but we tweaked it, we refined that for the purpose of a multinational consortium, because people use lithium in different fashions all over the world, and to come up with a phenotype is not an easy task, a phenotype definition. So we did that, we had a good inter-rater reliability on that. So this is what we then used, and we have the largest genome-wide association study of lithium response in the world, and we even got a genome-wide significant result. These are 4 markers. They're highly correlated, so it's more like 2. They are long non-coding RNA. It's not important what they do because we really don't know what they do, because but it's a proof of concept. With 2,500, we get a genome-wide significant result, because maybe genome-wide lithium response is something so homogeneous. The earliest studies on genome-wide studies on depression had 10,000 people, and they did not get a genome-wide significant result. Here with 2,500, we do get a genome-wide significant result. So it shows you that maybe depression is a very heterogeneous phenotype, but lithium response is much more homogeneous. That's what we can do through phenotype refinement, like a pharmaco-defined phenotype. So we published that in the Lancet, and it inspired a lot of folks. We have done trainings all over the world, and we hope to, hopefully one day, to be able to recruit also in Kiev. And we start in many other sites. So we have, these were the red ones, are the sites that participated. And now we are working on a new grant continuation to include more European samples, and hopefully also other sites where we held trainings or where we will start in the near future. So you can look that up. So Italy has been a very important site, from Sardinia, from Napoli. So we have very good colleagues contributing, and Italy has been also a very bulwark of lithium use. So I thank you for that. And but you also may be familiar with all these pharmacogenetic tests that are being sold, especially in America. When patients in America come and they come to us, then they have a result from one of these tests, and they say, hey, this drug you shouldn't give me, or I want this drug because the test says I need this drug. But this is kind of dangerous, because many of these studies or these tests are based on small samples, sometimes 200. Because you can go out and find someone who gives you a lot of money to come up with a company, and you come up with a test and say, hey, this gene is cool, I'll do a test, I have something in the literature that proves that this is interesting, and then you can come up with a test. And there's no regulation really, and so you can sell it. Of course, you cannot sell, you have to be careful in what you promise, but people, they believe that this may help. But I think, and we know actually, that there are so far only a few variants, like the CYP450 enzymes, that are, these are they look at metabolism. But even there, we want to check whether, in large samples, whether giving drugs based on genetics is actually better than just treatment as usual. So we have this European-wide consortium, SciPGX, where we have several sites in Europe. I also have one site here in the US, in Syracuse, where I live also. And so we want to do 1,000 people versus 1,000 people, 1,000 that are being treated based on the genotype, 1,000 that are based by treatment as usual. And we want to see whether focusing on these genes actually has an impact on outcome. But all the other tests that are out there that claim this gene is important and so forth, many of them there's no evidence. So beware of that. Just be aware of that, and be careful and educate your patients. Because what is really dangerous, if a patient says, this test tells me I shouldn't take this drug, how do we know that this person doesn't have maybe a variant that actually we don't know about, and maybe actually is good. This variant helps him, or this variant is good in combination with this drug. So there's so many things that are unanswered, and we need to study them before we sell these tests big time. So at the WPA, we try to promote this kind of thinking. We have held meetings in Kolkata, in Abu Dhabi, in Yerevan, through the section on genetics, educating people about pharmacogenomics. And there are many questions, and that's the WPA is a partner of this European-wide consortium, and this allows us to have educational sessions to not only spend like 10 minutes, but have an hour where we really interact with folks and discuss with clinicians, discuss the benefits and the prospects of pharmacogenetics. I also think we need to go multi-omics. That means include the metabolome, the proteome, the epigenome, the transcriptome. Just a quick glance, we just did a study on proteomics. We see different profiles between schizophrenia and bipolar disorder. We also did a large multi-ethnic study in Russia, Germany, Austria, and China on lipidomics, on lipids. So that's the largest study ever done on lipidomics. And we see various classes of lipids that show a differential expression between cases and controls. Not so much between the different disorders, but this is a first start. And we believe that lipidomic profiles may be at the center of health-related aspects like diet, lifestyle, ethnicity, genetics, epigenetics. So on many aspects of health and mental health. So I think this is a new avenue to pursue. And if you want to learn more about genetics, please check out the WPA section on genetics, or check out the section, the International Society of Psychiatric Genetics, who I used to be president of, and I'm still a proud member. We just recently published here, Mario Mai was so gracious to publish our paper here on education, on psychiatric genetics education in world psychiatry. So there are, I think, psychiatric genetics is an interesting area. There are a lot of promises, and maybe we have made many promises. We made many promises in the past that we haven't kept. So I tend to be very careful now, but I think it is something we cannot ignore. It's something we need to do. There's no way around it, and we have to not only do genetics, genomics, but the whole range of multi-omics. And with this, I'm done. Grazie mille a tutti.